Okay, so in this lecture, we're going to discuss the neuromuscular junction as well as the sarcomere. So the sarcomere is the micro level of muscle. So the macro level we would consider is just this entire arm, right? This is the macro level. At the micro level is when we start talking about the sarcomere and the neuromuscular junction. And so let me clarify what I mean by neuromuscular junction. So when I say that, what we are referring to is how the nervous system communicates with the musculoskeletal system, or more specifically, just the muscles themselves at the microscopic level. So if I'm at the gym, working out, lifting weights, in order for that to happen, the nervous system has to communicate with the muscles, right, in order for a muscle contraction to occur. So what a muscle contraction is, it's the shortening of the sarcomere itself, okay, at the microscopic level. And so in this lecture, what we're going to discuss is how that actually happens. And so if you look on the board, what you'll see is that at the very end, I have the part which is known as the cross-bridge cycle, or the power stroke. So the cross-bridge cycle is the movement, is the shortening of the muscle. So let me kind of just like introduce that so you see what I'm talking about, how we'll get into the details of it later on, but let me just introduce you what I mean by that. So if you look here, this is a single sarcomere. And so when I say it's shortening, the sarcomere is shortening, so when the muscle is in a relaxed position, or when the muscle is relaxed, it's going to be in this position. In the contracted state, it's going to move in this direction, towards here, towards the middle. Okay, so we're going to come back to this, back to the sarcomere. Okay, so the first model that I have here is this one, and this one is this model is the neuromuscular junction model. And so if you look at the model from this position, this entire thing here, so I'll turn it this way and you can see it here. So this is the axon. So this is the part of the neuron that's known as the axon. And then these are the axon terminals. So the axon terminals, they're going to synapse at this part. So this part is what we know as the neuromuscular junction, what I was discussing earlier. And so these little yellow dots here, these are what are known as vesicles. So vesicles contain a neurotransmitter known as acetylcholine. And so what will happen is an action potential will travel down this axon, so it'll travel down this axon, and then it'll reach the axon terminal. Once it reaches the axon terminal, calcium will enter, and that'll cause the release of our neurotransmitter into the synaptic cleft, or this part. So it's number 15 on here. This is what's known as the motor end plate. So you have the motor end plate. So acetylcholine will be released here. It'll bind to its receptor. And once it binds to its receptor, there's a flux of ions, and the flux of ions results in an action potential. So next week we'll discuss like the details of like what exactly an action potential is, but basically it's just neural impulses that are going from, it can go from neuron to neuron or from neuron to the muscle, which is the case here. Okay, so the action potential will travel along what's known as the sarcolemma. The sarcolemma is the fancy word for the cell membrane of the muscle. And so that's what's covering, if you look at it from this perspective, that's what's covering here on the outside. This is the sarcolemma. So you have the sarcolemma here, and then the part that's here on the outside, this is what's known as the endomysium. So the endomysium is what's going to cover the entire structure here. So all of these, so together, individually, these are all known as myofibrils. But as a whole together, it's known as a muscle fiber. So that's what it is as a whole. So what's covering the muscle fiber itself is, first off, the sarcolemma, as well as the part on the outside, which is the endomysium. Okay. So now let's move on to, I also want to point out the mitochondria. So that these individual things, these are the mitochondria in here. And if you look at it from this point of view, you'll see these lines here. So there's a red line here, as well as another red line here. So that's the boundary for the sarcomere. It's called the Z disc. So if you look at this, this is what's known as the Z disc, right? So this is this individual sarcomere, and this is the boundary of it. Okay. So we know what the sarcomere is on this model. And then let's look at some of the other components of the sarcomere and compare it to the other model. So this part in white is known as the sarcoplasmic reticulum. This is where calcium is stored. So this is the sarcoplasmic reticulum. And then this part in orange is known as the transverse tubules, or the T tubules. So if we compare this with this over here, it's the same. So this part in green, that's the sarcoplasmic reticulum. This part in yellow, that's the T tubules. Okay, so I'm going to take that back off. And then here on the sarcomere model, what we have is these thick lines here. These are known as the thick filaments, or it's also known as the myosin. So you have the thick filaments, and then you have the thin filaments, which are here. So these are the thin filaments. Okay. So let me point that out here on this model. So the part in blue here, this is the thick filaments. The part in red, those are the thin filaments. Okay. So now that we have a good understanding of what it is that we're looking at, we need to look at a couple of different of the regions that are found on this model. And so there's two different ways that you can look at it. So the part where it's the thick filaments only, that's this part here in the middle. So you have the thick filaments here in the middle, and then what's holding the thick filaments together is what's known as the M line. The M line. The M line is in the middle. So that's what's holding these thick filaments together. And so if we compare that with this model, so this part would be the M line, and the region where there's thick filaments only, that's what's known as the H zone. So the H zone is found here, and then the H zone is also found here in this model, where it's just thick filaments only. So you can look at it from this point of view, and then that's what it's also depicting for the individual myofibrils. So A is depicting the thick filaments only, which is the H zone. And then what's linking them together, so this is C, you can see that all of these are thick. That's the M line that's linking all of them together, which is what I just showed earlier. So you have this one, and then that I just mentioned, and then you also have these B, which is the thin filaments only. So the thin filaments only is what's known as the I band. So you have the I band there, and then I already mentioned this, and then part D is the A band, which includes thick and thin. So let's look at that on this model. So the part where it's just the thin only, which one is that? The I. Yes, it's the I region, the I band. This is the M line. This is the
H.
H zone. And the part where there are thick and thin is the A band. Correct. Excellent. Okay, so now we know all of the particular regions. Some of the other ones, just to cover real quick. So this part is known as the myosatellite cell. This is which is known as the nucleus. And yeah, that should do it for the basic parts of it. So now that I've already kind of like introduced as far as like action potential and what goes on for the neuromuscular junction. So now let's get into the details of how we get to the end, which is the cross-bridge cycle. Okay. So we've already discussed the action potential. We've discussed the release of the neurotransmitter here, which is inside the vesicle. The acetylcholine will bind to its receptor. We'll get the influx of, or the movement of ions in and out of the cell. The action potential will travel along what's known as the sarcolemma, or the cell membrane of the neuron. And then it'll travel, and it gets through these little pores right here. Okay, so it'll get through that pore, so that's number 14. And the action potential will travel here into where this part, which is known as the triad. So you have the triad here, and once that action potential reaches this position here where the T tubules are, calcium will be released by the sarcoplasmic reticulum, right? Because that's what I said earlier, that's where the calcium is stored. So once the action potential is sensed, the calcium will be released. And so the reason that we need calcium is because that's what's required in order for a muscle contraction to occur. You need calcium. And so we'll talk about where calcium goes here in a second. But once that action potential reaches here, the reason that calcium is released is because there's what we know as voltage-gated channels. They're like these little proteins that'll sense that change in the membrane permeability. And from there, that's what will cause the release of the calcium from the sarcoplasmic reticulum. And the part that's here is known as the terminal cisterna. So that's here, these little bulbs. I can get it closer towards the camera. That's these little bulbs here. So then, where does calcium go once it's released? So I pointed out the thick and the thin filaments on this model. So a couple of the other different components. So the part that's in red, that's what's known as actin. The part that's in green, that's known as troponin. The part that's in yellow that wraps around, that's what's known as tropomyosin. So I have this part taken out so I can show it here on the camera. So the part that's in red, the thin filaments, that's actin. The part in green, that's number 8, that's troponin. And then the part in yellow is the tropomyosin. And so why are we learning the different names of this stuff? Well, earlier I mentioned that calcium is released by the sarcoplasmic reticulum. And where it binds to is what's known as troponin, which is that part in green. So the reason that calcium has to bind there is because in order for the cross-bridge cycle to happen, which is the myosin head attaching to the actin, calcium has to bind to troponin. So once it binds to troponin, it unravels the tropomyosin. So the reason that tropomyosin has to unravel is because it's blocking the binding site for the myosin head, which is found on actin. So the myosin head has to bind to actin, but the only way it can bind to actin is by unraveling the tropomyosin, which is initiated by calcium. So once this process is initiated, the myosin head binds to the actin, and then it brings it closer towards the M line. Therefore, we get our muscle contraction. Okay. So that's the process as far as the the cross-bridge cycle. But what the power stroke is, so have you heard of rigor mortis? Yeah. So when people die, a lot of times the muscles, they'll become real stiff. And so the reason that they become real stiff is because the myosin head remains attached to actin and the muscle is shortened. So the muscle will remain shortened because if they're dead, there's no ATP available. And because we need ATP in order for the cross-bridge cycle to occur. Because whenever the myosin head is attached to actin, we have ADP and inorganic phosphate, which are attached. But then in order for it to come off of it, you have to have enough ATP. So if there's no ATP available, the myosin head is going to remain intact, and therefore you get rigor mortis. Okay, so that's going to do it for this lecture. Are there any questions?